In this chapter, we are going to be discussing some basic ideas in genetics. So on to the review overview slide. So we're going to talk about some key terms in genetics that are very important for you to understand this process. Also, we're going to be talking about the typical Mendelian experiment. You must understand this experiment. In genetics circles, people that study genetics, any kind of genetic publication, they will refer to the typical Mendelian experiment. You're just expected to know that like it's your last name or your first name. So it's a very important understanding or a series of crosses among individuals. And I want you to have a good understanding of how that experiment flows. And then also something along with that typical Mendelian experiment that's very important is something called a testcross. Make sure you understand what a testcross is. And then also looking at multi-hybrid cross. So looking at more than one characteristic at a time. In a typical Mendelian experiment, we're just looking at one characteristic. You might remember purple versus white flowers. We'll take a look at those classic examples from Mendelian genetics, but also in the multi-hybrid looking at more than one characteristic. So maybe color of flowers and the color of the seeds, okay, at the same time. And then finally something called pedigree analysis, where we look at human characteristics and try to predict characteristics in offspring. All right, so some genetics vocabulary. Here we have our pair of homologous chromosomes. As you notice, the sister is not attached here, so we aren't getting ready. We're not gearing up for any kind of a division process. And we have the genes are located along the length of the chromosome. They're indicating one gene. And different forms of a gene are called alleles. So for instance, in the pea plants, we have two forms of the allele for flower color. One is a purple and one is a white allele. And depending on your parents, you get either the purple and a white, you could get a white and a white, you can get a purple and a purple, so all different kind of combinations. And those combinations result in the genotype of the organism, which we'll talk about here in the next few slides. But with typical or classical Mendelian genetics, we have a complete dominant situation where one allele is dominant over the other. So if we have one of those dominant alleles, for instance here in flower color, purple is dominant to white, this flower, if it has one purple and one white, will be purple. If it had a purple and a purple, it would be purple. You couldn't really tell a difference in this particular inheritance pattern or allele combination. And the only way to get a white flower would be to have two white alleles. Okay. Now, looking at the genotypes, what kinds of alleles the organism has, if they have two of the same kind of alleles, they're called homozygous. Homo meaning same, and they have the same two of the same alleles. So for instance, like we had said, two purple flower alleles. We indicate the dominant allele in Mendelian genetics with an uppercase P. P for purple, we always name it after the dominant allele. And we have two of them, so we have uppercase P. And we say that this organism is true breeding, that because they can only produce gametes, egg or sperm, when they go through the process of meiosis, that have purple alleles. That's the only thing they can produce. Almost like a purebred dog. Okay, true breeding. Now, if they have one of each, they're called heterozygous. One of each. There's only one genotype for that. Now, again, with typical Mendelian genetics and complete dominance situation, that P, that purple allele, is going to dominate over the white. So this flower will look purple. It will look exactly the same as this purple flower. Okay. All right. So again, the genotype is what are the alleles, and then something called the phenotype. What does this organism look like? That is the phenotype. The phenotype is always based on the genotype. So here are the three genotypes. We have heterozygous. This guy's going to be purple. We already talked about that. Homozygous dominant, purple. Homozygous recessive, white. Okay. And this chart just reviews that situation. So again, we see that the homozygous dominant and the heterozygous individuals look exactly the same. We'll talk more about that in just a bit. The typical Mendelian experiment. Here it is, my friends. Here it is. So in a typical Mendelian experiment, we always have something called the P or parental generation. We must have true breeders. So this, we know a true breeder only can produce one type of allele for whatever trait we're looking at. So we'd have to have a dominant allele and a dominant allele, homozygous dominant. We'd have to have two recessive alleles, homozygous recessive for this white flower. Okay, we cross these two. The way that Mendel crossed or cross-pollinated his pea plants is shown in the chapter. So if you're interested, please take a look. And how he produced his generations. And this first generation is called the first filial means son, generation, F1 generation, and they are hybrids, meaning that they have one purple allele and one white allele from the other parent. And we have our F1 generation, and what happens here is now these organisms are all cross-pollinated with one another. So there's more than one plant that's produced, and in doing so, we have all hybrid parents for our F2 generation. And as a result, what he saw was he'd always get through a 3-to-1 ratio of purple to white phenotypes. So he took a look at the genetics, and he came up with the idea of this dominant versus recessive based on his experimentation with pea plants. And if we take a look at the alleles, you can see here the only type of allele from the purple flower is the dominant allele. The only type of allele from the white flower is the recessive. And we have our F1 generation. We have one of each. And we can make something called a Punnett square for our F2 generation because we have a heterozygous parent flower, another heterozygous parent flower. And if this sperm combines with this egg, we'll get a purple homozygous dominant flower. The same goes for all the others. This sperm with this egg, we get a heterozygous purple flower, and so on and so forth. And there is the 3-to-1 ratio of phenotypes. Okay, so be very familiar with the Punnett square. If given parents, you put one parent on one side, another parent on the other side. You've probably seen this before. Okay, so nothing too new here. Now the only thing that goes along with this is, well, we need those true breeders for our parental generation of a typical Mendelian experiment. How do we go about doing that? How do we get those, for sure, get a true breeder for that purple flower in the parental generation? Again, we know that the white flower has to have that genotype, little p, recessive, in order to be white. So that's not a problem, true breeder. But that purple flower, remember, we're not sure. We don't know. Is it homozygous dominant, which we would hope it would be, or is it heterozygous? So is the cup half full or is the cup half empty? I'm pretty much a cup half full kind of person, so let's go over here first. Let's say that the purple flower is homozygous dominant. We cross it with the white flower. So this is a test cross, one of the test crosses. A test cross, you take an organism of unknown genotype, cross it with an organism of known genotype. We don't know the purple flower, but we do know the white. And we should see all purple offspring if the purple flower is homozygous dominant. Now, if the cup is half empty and our purple flower is in fact a heterozygote, we would expect, and we cross it with that purple flower, we would expect to see about 50-50 ratio of purple to white organisms in the offspring. Okay. So we could use this purple flower over here. Can't use this one for that typical Mendelian experiment. We're going to stop here and pick back up with multi-hybrid cross in the next